these next two talks are sort of get you prepped for what you're going to do tomorrow in the lab. This first talk will give you sort of an overview of the workflow and how things are done on the Ibis T5000. It's actually 10 easy steps, which the next slides will go over what we're going to do. The first thing you want to do is when you order, you will receive a kit, and upon receipt, you want to make sure that you have all the assay components. That includes your barcoded assay plates, magnetic bead plates, elution plates, your cleanup reagents, an instruction sheet on how to use all of these, and then a CD with the import file. So, unfortunately, all of these components require different storage conditions, and so you need to be aware of that. Oops. For the assay plates, they need to be stored at -20 degrees C in a manual defrost freezer. For the forensics kits, the assay plates will contain everything you need except for your template. Each well in the plate has 35 microliters of PCR master mix, which includes the primer pairs, the modified DNTPs, the PCR buffer, and the enzyme. As Tom mentioned in his talk, each column is a separate sample with 8 wells comprised of 3 primers per well, and your profile is a composite of all these wells. You'll need to add 5 microliters of template to each well of your sample, so you'll need somewhere around 50 microliters of your sample to complete the assay. The magnetic beads, on the other hand, must be stored at 4 degrees C, and in fact, if you freeze them, you will ruin them. And we request now, because of shipping, we've had problems with the mag beads going one place and the liquid going to the top, and then your mag beads dry out, and then they don't function, that upon receipt, that you centrifuge them for 15 seconds at 800 RPM, and then put them in the freezer in an upright position so that your mag beads are covered by the liquid. The elution plates are your simplest. They're just empty MJ research plates with a barcode. You can store those at room temperature. The cleanup reagents, while not in use, we also recommend storing at 4 degrees C, and you must add methanol before use of the reagents. On the bottle is the volume to add and a nice little checkbox for you to indicate that you've added methanol. And we recommend the HPLC gray from Burdick and Jackson. And also, which most of you would do anyway, we recommend tracking lot numbers of the reagents so that if you ever have a salt issue or something, you can call us up and say, I was running lot X, my plates are salting. We can go check our internal QC and pull that lot and check and help you troubleshoot where the salt's coming from. So your next step is now you have all of your assay plates, but you have to have a way to use these barcodes, and so that's where those import files that are sent on the CD come into play. The Ibis Tiger Track has an import wizard in which you would import these files. When you first receive kits, you receive a plan file which tells you what the assay is and tells the instrument how to use it, what the sample layout is, where the primers are, and all of those things. You typically just need to import this once, and it will have plan in the title. The ones that you need to re-import each time are the barcode files, and they'll have Ibis kit in the title. Depending on how the kit is assembled, you can have individual files or one big file that'll have the barcodes for the assay plates, magnetic bead plates, and the elution plates. If you don't have these plates imported, you can't register your samples and you can't run the T5000. It requires that these resources be imported. So the next step is upfront sample processing. In the service lab that I run, we're lucky in the sense that we get a lot of extracted DNA, so I get to skip this step all of the time. One of the nice things about our system, our PCR plates are made to handle a variety of extraction methods. So you can do QIAGEN columns, various magnetic bead-based systems, phenylchloroform, and many others will work with our PCR plates. Then the next step is registering your experiment in Ibis Track. There are several wizards that have been designed to help you set these up, and the one you choose will depend on the number of samples. And this just requires that you have a sample list in a Microsoft Excel spreadsheet format. And you can register to set up plates from tubes or from a plate. So you have great variety. The other thing that's nice in your sample list, you just need to have your samples. Your assays are designed to work with control layouts, and it's your control layouts that will tell you where you're going to put your negative PCR controls and your positive PCR controls. And they'll interleave those among your samples automatically for you. The next step is setting up the PCR plates once you've registered them. You can either set them up by hand, which I don't recommend, as some of the people in the room have gone through the training and have done it by hand. If you haven't done it in a while, you will make some colorful expressions. We recommend highly using a robotic setup. We have a lot of experience with the Janus robot, but other robots can be used as long as they can handle doing 5 microliters dispensing. 6 through 9 are kind of the easy steps. 6 is just the heat seal. And the reason we make a point about this heat seal is this is a heat seal that is important for use with compatibility with Ibis T5000 downstream. If you use plastic heat seals or some of those wax heat seals, you would break the T5000 and you'd be making a service call. Then we thermocycle the plate. Then we prep the T5000 for use by filling reagents and emptying waste as needed and loading your plates in the hotels. And then basically you hit the start button and run your system and walk away. So the next day you would come back and you would analyze your data using the software that Tom described. So that's sort of your 10 easy steps. So how much time do each of these steps take? Well, you guys are familiar with your sample prep. We know that with a Kingfisher in about an hour with 30 minutes of setup time and run time, we can do 96 samples. Plate setup can take anywhere from 10 to 20 minutes manual or 15 minutes per plate with a Janus. And again, we recommend the Janus because it's more reproducible than the human and less frustrating for the human. Right now, the mitochondria assay, which is our current marketed kit, has a 3.5 hours cycle time. The T5000 that's commercially available has these time ranges. You have a 20-minute startup time, which involves system flushing. Then it takes about 45 minutes to clean up a plate. After the first plate, this is all done while other plates are spraying. Then you spray one plate every 90 minutes. And then also in parallel is data processing, which takes 15 to 20 minutes. So if we were to just start with extracted DNA, which is what we mostly do in our lab, here's what your day might look like. On day 1, you're going to be able to register, set up, thermocycle, and load your Ibis T5000 for easily 9 plates or more. The samples we're processing right now come as 91 samples, so we think in terms of 9 plates. Then on the second day, you do your data analysis and reporting. Your data analysis can take from 15 to 30 minutes per plate, and time will go down as you are more experienced at analyzing the plates. And then generating the report that Tom showed you takes less than 5 minutes. So if you were to make some assumptions that you're doing manual PCR setup and you only had 2 thermocyclers, in an 8-hour day, you could do 4 plates a day, which is 40 samples a day, and which will get you to about 10,000 samples per year. If you do robotic setup and bump up to 5 thermocyclers, you can easily do 10 plates in a day, and that gets you to 26,000 samples per year. So one of your limiting factors in how many samples you can do is how many thermocyclers you have, and that's why I have 24, so I can load them all up and crank through them and going through them. So you can see that you can get tremendous sample throughput with this. And this this is 100 samples in plus the positive and negative controls that you do. So you're actually running more physical samples, but I just gave you the number of samples you can do per day of actual work. So that's sort of the general overview of the workflow that goes on in our lab, in which you guys will sort of get exposed to tomorrow.